Hello all. I want to start off by thanking Raphael and Vince for putting together this great educational session. My talk today is going to focus on evaluating different sources of variation in functional brain network measurements and the use in particular of precision or extended sampling fMRI approaches as a way of doing that. This is something that I'll focus on in the second half of my talk. I'll start off by noting that our ability to map functional brain networks with fMRI has really advanced a lot in the past decade, allowing us to create these really beautiful and detailed maps, much as you can see here for a group average of 120 individuals at rest. And we've now shown that with sufficient data, we can also map these networks quite reliably at the level of individual participants. I'm not going to overly belabor these methods since I'm sure you've been hearing a lot about them over the past few talks, but I do want to introduce a few important ideas that we're going to come back to at multiple times during this particular session. The first, of course, is that when we talk about functional connectivity, what we mean is measuring some level of statistical association in the activity patterns between different brain regions, often through a correlation-like measurement. So for example, we tend to find that the bold activity between regions that are part of the same system, like the motor system, tend to be highly correlated, whereas the bold activity between regions that are part of different systems, like a visual and a motor region, tend to show much lower correlations. And of course, what's intriguing is that this is true not just when we ask people to complete a motor task, but even in the spontaneous activity patterns that we find during a resting state. The correlation patterns during rest of motor-related regions mimics the patterns of activity we might see in a motor-related context. And of course, this is true not just for 2 or 3 regions, like I showed you on the previous slide, but these measurements can be made for many different regions spread throughout the brain and plotted all at once in the form of a correlation matrix, or in the parlance of Olaf Sporns, a connectome, where any given cell in this matrix just represents the correlation between a pair of brain regions. Now here I've ordered them randomly, so the pattern looks a bit like snow, but if I reorder these regions such that I put those that are part of the same system close to one another, you see this really beautiful structure emerge, where you can see high correlations along the diagonal, indicating regions that are part of the same system, and low correlations in the off diagonals between regions that are part of different systems. So this nice block diagonal structure allows us to then effectively use different types of community detection algorithms to identify systems of the human brain. Here we used InfoMap. I'm happy to talk more about that in the question session later if anyone is interested. And I'll just mark off the different networks with red lines as shown here, or I can paint them on the brain in different colors as shown here. So this is a really powerful approach to identify in a data-driven way the systems that we find for the human brain. And even though it seems relatively unconstrained, this approach has been shown to be quite robust. Just as one example, I'm showing you here group average representations of the human brain systems that have been found at WashU on the left and on a separate set of individuals at Harvard on the right, using 2 different clustering techniques collected on different scanners. And yet you can see that there are a lot of commonalities in the systems that have been identified across these 2 data sets. These methods have generated a lot of interest for myself, and I think for many of my colleagues, in our ability to use functional network measurements to address many different types of questions. For example, how these networks are altered with different forms of brain damage and disease, how they may differ across people and perhaps relate to individual trait-like characteristics, how they may change over the course of the lifespan with developmental or aging processes, or how they may change under different contexts with different demands of our current environment. And indeed, we can think of functional networks as potentially having variation on multiple different ends of the spectrum on a full continuum. On one end, you might hypothesize that these measurements should actually be invariant, that everyone should have more or less the same functional network architecture, just as we have more or less the same body plan, and that the only differences that we're measuring are due to sampling variability or measurement noise. On the other extreme, you might think that these networks should be constantly changing as we engage in different thought processes and our neurons are firing at different rates. But of course, they may also change at intermediate timescales over the course of minutes, for example, when we enter into different states of arousal or different task states. over the course of hours or the day, for example, with circadian rhythms, only after extensive experience in particular context or with, say, longitudinal developmental demands, or that they may be fundamentally stable within an individual, but very meaningfully across different individuals. And so, of course, I'll just sort of reflect back on the previous slide to say that depending on the characteristics of these functional network measurements, that may make them better suited for addressing different questions that I posed earlier. For example, you may want a relatively stable measurement to track aspects of a person's disease status or traits at other moments in time, but you may want a very malleable measurement if you want to track ongoing context. So in my talk today, I want to reflect on these ideas a little bit. In the first part of the talk, I'm going to tell you about some of our older work looking at evidence for and against stability in functional network measurements. And then in the second part of the talk, I'll tell you about some of our more recent work using these precision fMRI techniques to contrast the relative magnitude of these different sources of variation. Before I get started on the actual work, I want to give some qualifications for the data that I'll be talking about today. So all of the data sets that I'll be talking about are based on fMRI, and in particular the bold signal, which is, of course, relatively slow. Most of the power in the bold signal is below 0.1 hertz. And so these factors, the variation may not extend to other types of measurements like EEG or single unit recordings, which are going to capture very different timescales of neural processing and may have their own different statistical properties. I'll also sort of define here that when I talk about time varying functional connectivity or dynamic functional connectivity, what I'm talking about is variation in the functional connectivity matrix that is in the second order statistic of this connectome, not in the original activation time course, which is of course a first order statistic often used to track invoked responses. So with that, I want to get started on evidence that there has been on whether or not functional connectivity changes across states, such as periods when you're engaged in a particular task or changes in arousal. And of course, many groups have studied this, for example, contrasting functional connectivity during a task period with rest. Just showing you here one example from some of our own work where we can show that when you take the direct difference between task and rest, you can find these significant differences in functional connectivity, both within different brain networks as well as between different brain networks. And in fact, we can also show that these differ depending on the specific task that someone is engaged in. You can see here examples from a semantic task, a mental rotation task, and a dot coherence task. We can see that there are different patterns of functional connectivity across these conditions. And indeed, a number of other groups have used this sort of functional connectivity data during task periods to show that you can effectively decode which task a person is completing from their functional connectivity profiles. And I'll close here by noting that significant variations in functional connectivity structure can also be seen with more subtle changes in state, such as those associated with opening or closing of the eyes. So next I want to turn to using these sort of observations to interpret functional connectivity changes that are seen during the resting state. When we subjectively think about the resting state, we often think about all the different types of things that any given person can be thinking about inside the scanner. And we might reflect that that could be observed in the resting state correlation patterns as well. And so I'll just start off by noting that this is a project that's actually led by Tim Lauman, on which I collaborated. So I'll be talking a little bit about his project. And so one of the ways that dynamics in functional connectivity have been measured during rest is by using a combination of sliding time window techniques with some form of clustering, such as k-means clustering, as shown here. And so this might allow you to observe whether functional connectivity matrices made on different small windows of data might vary meaningfully with one another. And the results of these past studies have suggested that there might be additional structure to functional connectivity at rest that may accompany our subjective feeling of changing thoughts during this time window. However, care needs to be exerted in the measurement and interpretation of dynamic functional connectivity, because unlike those task-based modulations I showed you earlier, we don't have that external imposed perturbation that we can use as sort of our standard of trying to understand which variations are meaningful and which are less meaningful. And of course, there are many other forms of intrinsic variability and sources of noise that can also affect functional connectivity data. And so I'll talk a little bit about these different forms of noise and what you might watch out for in terms of interpreting your own data. The first that I'll talk about is sampling variability. By sampling variability, I'm just referring to the variability that you probably learned about in one of your first statistics class. That is that your sample, your particular piece of the time series, is just an estimate of a statistic from the full population. And this sample estimate will have a certain amount of variability or error associated with it. And this type of error tends to go down with increasing samples from the population. Now, this is true of any given statistic, but the same has been shown to be true of functional connectivity measured over time, which shows a clear progression of decreasing error with additional time points added to the sample. Since dynamic functional connectivity is typically measured with a very small amount of data, we need to be particularly careful about this type of error, which can make it look like there are large variations in the time series simply because there are variations in the error over time. So how might you account for these patterns that may appear dynamic but could be due to sampling variability? One way is to construct an appropriate null model that is fully stationary but includes some noise, and monitor whether your actual data shows any differences relative to this null model. And I know you'll have heard a whole talk about how to construct the appropriate null model for a given question before from Raphael. I'll just tell you a little bit about the particular model we used in this project in order to contextualize the results I'll tell you about later. And so Tim's idea was to create a null model that was stationary, that is, had a static functional connectivity structure with noise, mimicking bold data, but didn't have any true dynamics over time. And then he contrasted real data with this stationary noisy model. So he started off with Gaussian noise, and then he imposed the stationary network structure and also imposed that it should have the same power spectrum that true bold data has. And he contrasted this stationary simulated model with the true data from functional connectivity measured with fMRI. You can see examples here of the simulated and the real data, and I would probably argue, if you let it play out for a few moments in time, that it might be hard to pick out which of these is which just from these movies. You can, of course, put up the titles here so you can see them afterwards. And in fact, you don't just have to take my word for it. If we do different statistics on these two different data sets, you'll see that they are also quite a bit like one another. Indeed, if you apply a sliding time window and k-means clustering analysis to the simulated data, you'll find very similar states as what you find in true fMRI data, suggesting that states on their own are present in both real and stationary simulated data sets, such that the presence of states can't be taken as proof that the data is indeed dynamic. I'll put up a link here to Tim's model. So if you're interested in incorporating this model or using it to compare to your own data, you can go there and use it to generate similar models relevant to your own work. So that's a simple form of variation that we should be concerned about. But of course, there are other more systematic biases that are also true in functional connectivity data. One of which might come to mind is motion, as its transient nature may make it look a lot like a dynamic process. When we look at it more closely, we can show that motion does indeed correlate with the kurtosis of the bolt signal, that is the variance of the covariant structure. And so what you can see here is the mean FD of a given session correlates with its variability and functional connectivity structure. Once you have removed those high motion windows, you can find that this variability is drastically reduced. Note that Tim didn't look directly at respiration-related effects of the bold signal here, but respiration, especially related to transient deep breaths, may also induce transient patterns in functional connectivity that can inflate dynamic characteristics. Moreover, the relatively small amount of remaining variation, note the change in scale here, was shown in this data set to be related to arousal levels across different scans, such that having lighter sleep led to having higher kurtosis. And so this suggests that other forms of states, such as sleepiness inside the scanner, can also contribute to dynamic functional connectivity measurements. I want to mention another project that's being led by my graduate student, Zach Ladwig, which was inspired by recent work from Farnaz Esfalani and Rick Betzel and colleagues looking at these intriguing apparent events in functional connectivity. These are intriguing moments that appear to represent times where co-fluctuation between different brain regions is particularly strong and represent moments that have very strong modular network structure relative to non-events or periods with particularly low functional connectivity associated across regions. These led the authors to argue that these may be transient moments that drive functional connectivity. Zach conducted a number of analyses to dig deeper into the question of whether these transient events do indeed drive functional connectivity or if there may be other explanations for these results. I'll only tell you about one of these today, which is to say that he created a similar simulation, similar to the one I just told you, in which he took a static network structure and imposed Gaussian noise on it, and asked whether or not some of the event properties would be present in this stationary simulation. And with this data, he found that this fixed but noisy simulation recreated event-like behavior similar to that seen in real data, suggesting that a simple explanation is that events can be a natural outcome of sampling variability in fMRI. Now, if you're interested or want to dig in more into this work, I'd encourage you to check out more details at Zach's OHBM poster listed here later this week. So here I've shown you evidence that functional connectivity shows significant modulations across different states, but also that variations in functional connectivity within a single resting state can be easily confounded with other factors, such as sampling variability, motion, and arousal, making it difficult to find resting state variation that might be linked to cognition. Moreover, there's also evidence that I introduced at the start of the talk that functional connectivity can show a strong degree of invariance across large groups. And so you might be asking yourself, how do these different contributions, how do these different effects contribute to functional brain networks? How do they contrast with one another in their relative magnitude and their distribution? And of course, this will then feed back into our original question of how we might use these functional brain network measurements effectively to answer different types of questions. And so this is what I'm going to turn to in the second half of my talk, where I'm going to introduce the use of the Midnight Scan Club dataset to address this question. Now, this Midnight Scan Club dataset was a dataset spearheaded by Nico Dosenbach, Steve Nelson, and colleagues at WashU. And it is openly available for anyone who's interested. The dataset's on OpenNeuro, and the code is available at the GitHub link I have below. And it's an intriguing data set in that it contains a lot of data for a smaller sample of participants. So 10 participants came in every day at midnight to complete 10 separate fMRI sessions. They were completed at midnight because the scanner was discounted 90%, allowing them to be this data set to be collected at a fraction of the usual cost, which may appeal to many of us young academics in the audience. This is one of a growing sample of data sets focused on collecting large amounts of fMRI and phenotypic data from a smaller sample of participants relative to typical fMRI studies. A growing trend for providing deeper or higher precision individual data. And this higher precision individual data has been shown to produce much higher test-retest reliability relative to more standard approaches. Now, the reason the Midnight Scan Club dataset is really well suited to addressing the question I posed is that it includes a lot of data, not only from resting state on each day, but also from 4 other tasks that were completed on each day. A semantic task in which participants were asked to complete a noun-verb judgment, a dot coherence task in which participants were asked to say whether dots were rated concentrically on the screen, a motor task in which participants were asked to move their hand, foot, or tongue, and an incidental encoding task in which participants were asked to categorize faces, scenes, and words. So this design allows us now to look for factors that were either common across this full group of data sets or selective to individual people, to individual sessions, or to individual task states. So for this experiment, we measured these correlation matrices or connectomes separately from each person, each day, and each task state. Each row here just shows you a couple examples of the high quality data sets that we obtained from these participants. And I'm only going to have time today to show you one way that we looked at the relationship across these, which is in the form of a similarity matrix, where any given cell in this matrix now represents the similarity between a pair of connectomes. I'll just introduce you to what you might expect to see in this matrix. If there were factors that were shared across the full group of data sets, you might expect to see high similarity across this full matrix. If there are factors that are shared by single individuals, you'll see the high similarity along this main block along the diagonal. Factors specific to a given task will be seen in this little mini block along the diagonal, and the off diagonals relative to that. And of course, you may get interactions between these effects. For example, factors shared by an individual and task in the single mini block along the diagonal, or individual and session in this sort of checkerboard pattern. So if we return to the actual data, what we ended up finding was a mixture of these different types of effects. You'll notice, for example, that the background is not centered at 0, but rather around 0.5 or 0.6 correlation, indicating that indeed there are some commonalities in these functional networks across all of the participants and data sets included in this project. However, relative to that group similarity, there's also this really prominent effect along the diagonal, showing that a given person's brain network measurements look most like themselves, regardless of what day or what task they're completing. There was a small effective task. For example, people look more like other folks doing the motor task if they themselves are doing the motor task, but it's a bit hard to see on this color scale. Now, relative to this individual effect, you might also notice a little bit of the small block structure along the diagonal, indicating that there's some interaction between individual and task. A particular person looks most like themselves doing the same given task, say doing the motor task with other days of doing the motor task. There is also a small effect of day-to-day variability, but again, it's a little bit tricky to see on this color scale. So I want to make 2 points here. First, much as past data sets have suggested, we find evidence for all of these different factors affecting functional connectivity structure. And indeed, they're already significant with just the 10 participants that we have here in the Midnight Scan Club dataset. However, if we contrast them with one another, we'll notice that they're quite different in their magnitudes, with most of the variation really dominated by group commonalities and individual specific features, much smaller effects driven by task state or day-to-day variability. Interestingly, the largest of which actually interact with individual differences. Also note that while the effects I showed you on the previous page were for the entire cortex at once, we also looked at whether these differed by different locations. We looked at this in a few different ways. I'll only tell you about one today, which is to say that we separated out networks that were involved in more basic sensory motor processing, like the visual, auditory, and somatomotor networks, from those involved in more top-down control, like the cingulo-opercular, frontal-parietal, and dorsal and ventral attention systems. And so when you look at the patterns separately for these systems, you see the same general trend, but you will find some significant differences, where individual specific effects, shown over here on the right-hand side of the plot, tend to be larger in control than sensory motor processing systems, whereas cross-subject effects tend to be larger in sensory motor processing systems than in control systems, consistent with past results suggesting there's more individual variation in these higher level systems. Also, I want to return to the qualification that I made at the start of the talk, that what I'm discussing today is about these variation in functional connectivity, these second order statistics, rather than in the first order evoked activity patterns. And indeed, if you made a similar analysis of activation maps across these data sets, you would see a different pattern with much more sensitivity to task state, but intriguingly, quite a large variation across individuals as well. Moreover, I want to go back to the point I made at the beginning about these properties and studies I told you about today as being fMRI specific, which of course is slow. An interesting recent paper from Farham Mostame in Sepidesarigiani's lab has suggested that this high level of stability in functional architecture across states may also be true of phase and amplitude coupling connectivity estimates from ECOG data, even when measured at much higher frequencies. So to summarize here, while all of these forms of variation exist in functional connectivity as suggested by past data, these results do show that functional connectivity data is dominated by stable factors, either commonalities across the group or stable individual specific features. Moreover, while we do see significant differences from day to day or across task states, these tend to be relatively small in nature. And finally, these effects are relatively consistent across the brain, but do show some differences, with group commonalities being particularly prominent in sensory motor processing systems, individual specific features being particularly prominent in top-down control systems, and state level effects showing a combination between the two. I want to close with some thoughts on the conclusions that I think we can make based on this data. The first is, I think it suggests something about the potential neurobiological contributions to these fMRI functional network measurements, suggesting that they're more likely to be driven by stable or very slowly changing processes rather than ongoing cognition or very transient neural firing. This in turn tells us something about how we might interpret functional network comparisons between groups and states. In the past, for example, there's been a lot of concern that when you, for example, compare functional connectivity between older and younger adults inside the scanner, that the differences that you measure might be due to differences about what those two groups are thinking about at a given moment in time. These results suggest that's much less likely to be true, that these effects are much more driven by these sort of slow or stable properties that are different across individuals, and that indeed, if we're interested in those state-based effects, that we might want to carefully account for those stable features. My group is very interested in task state effects on functional networks, and in fact, we've been using these sort of techniques to try to improve our ability to measure these task state effects. In a recent project led by graduate student Alexis Porter, she's shown that you can build classifiers that respect or take advantage of these individual specific features and use them to well outperform classifiers that are built to predict task state across different individuals. For example, using more classic leave one subject out classification performance. And finally, I want to leave you with the thought that these approaches may be exciting for how we can develop functional network-based methods for different medical applications, for example, in the development of biomarkers or neurosurgical planning. And I just posted up here a reference to another project that Alexis is leading, in this case, looking at specific forms of functional network variation that are seen in individuals with schizophrenia relative to healthy controls. And if you're interested in those ideas, I would encourage you to check out her poster that she has here at OHBM in the Wednesday, Thursday slot as marked below. So this brings me to the end of the talk, looking at evidence for and against stability and the relative magnitude of these different forms of variation in functional connectivity. And so I want to hopefully give you a few takeaway points building on other work earlier in this session. That first off, when we're doing work on dynamics or variation in functional connectivity, it's important to define our terminology and our constructs clearly so we can communicate well with one another. Secondly, that in this work, especially when we're looking, for example, across resting state, that it's important to address other potential sources of variation. This includes variation from measurement noise or sampling variability, perhaps through the appropriate null model, either Tim's model that I introduced here or one of the models that Raphael introduced in his talk. That's important to also account for systematic biases that affect functional connectivity, such as from motion or respiratory effects, with the appropriate denoising strategies. The references I have here do a nice job of comparing across many different pre-processing strategies that have been suggested to use to address these different forms of variation. And I would also like to bring up that we should account for the fact that arousal may be one of the forms of neural based changes that can systematically change functional connectivity architecture, and that we should account for this either through our measurements in carefully accounting or trying to remove arousal-based variation, or include these in our interpretation of dynamic changes that we may see in functional connectivity architecture between different groups. And I'm just putting up a reference here to a nice paper from Talyazuki and colleagues showing how you might measure arousal-based changes in a number of different public resting state data sets. And finally, I hope my talk has suggested to you that it might be valuable to collect substantial amounts of data per subject when you're trying to measure different forms of variation in functional connectivity. Partly because I think it gives you a nice picture of individual variation in these forms of functional connectivity, and also because it can provide a very stable baseline from which to measure other forms of dynamics. So if you're interested in this, I'd encourage you to check out a couple of tutorials that I've created, one in MATLAB and Python, they mirror each other, that introduces you to the MSC data set and how you might create some of the results that I showed you in this talk. And in fact, I might suggest that you might consider embracing the strong stability of the data and using it as a tool in your other projects to improve functional neuroanatomical correspondence across subjects, and thereby address some of the different forms of interesting or uninteresting variation to you in your projects. And if you're interested in this idea, I would encourage you to check out this recent work in the lab that will be presented by my research tech, Ali Doretsky, below. And you can see that in the Wednesday, Thursday session as well. And so finally, I want to leave you with a few reflection questions that may be relevant to consider when embarking on your next project on variation in functional connectivity. For example, how is this variation related to ongoing cognitive phenomena, arousal, or other neurobiological phenomena? What are the uninteresting causes of dynamics that could be present in your data set, and how can you deal with them or address them in some way? And how can one demonstrate the reliability and validity of dynamic functional connectivity, and what is the appropriate model for your particular dynamic functional connectivity question? I think this last one in particular is a fairly open-ended question that many of us still struggle with today, and is an interesting one to reflect on as you're working on these projects. And so I want to close by thanking my collaborators on these projects, in particular, Tim Lauman and Steve Peterson, who led the initial dynamics work I talked about in the first half of my talk. And of course, my current lab that's carrying on much of the recent work. And I'll go ahead and put my contact information up here in case you want to reach out with any questions you have that you don't manage to address at the end of the session. I'll point out that my lab is going to be moving from Northwestern to sunny Tallahassee at Florida State, and I'm recruiting particular postdocs for the lab, so do please reach out if you're interested in that sort of position. And I'll encourage you also to check out some posters that we have at HBM in the Wednesday, Thursday sessions coming up. Thanks very much.